Hello, I'm Professor Brown, and I'm going to review how to do a quadrant streak. So to do a quadrant streak, you're going to first of all need, of course, bacteria that you're going to transfer to an agar plate and your inoculating loop. And so I'm going to start with by sterilizing my inoculating loop. And I'm going to flame it as usual from the base of the loop towards the tip. And I'm going to get some bacteria. I have some E. coli here. And again, just get a tiny amount. I'm going to cool the loop on the agar and then touch and get a very small amount of bacteria. And I'm going to take my agar plate. Notice how I've labeled my agar plate. Name, date, and what's on it. So this is going to have E. coli. So I'm going to place it so that the agar side is down and the lid up. And I'm going to hold the lid like this. And then so notice I took the bacteria on the loop and streaked back and forth on one side of the plate. And I wasn't talking while I was streaking, because that's a bad habit. If you talk when your plate's exposed, you're going to get little microscopic drops of saliva and whatnot and contaminate your plate. Now, what I've just done is I've taken some bacteria from our E. coli slant and I've transferred it and done a little streak on the surface. Now, keep in mind the whole point of a quadrant streak is to get the bacteria on the agar surface dilute enough so that you get isolated colonies. Okay? A colony is, of course, a single bacterium or a single cluster of bacteria that ended up on the agar plate surface and overnight grew into a colony. So it's basically a pure culture. So if you want a pure culture, you need to get isolated colonies. This is a technique to give you nice isolated colonies. So there's actually a lot of bacteria in that streak. We want to spread them out. We're going to spread them out by streaking 3 more times, each time making the bacteria on that plate more and more dilute or spread out so we can get some isolated colonies. So since I've been talking too much, I'll flame this again just to make sure it's still sterile. And this is a key step here. I'm not going to get any more bacteria. All the bacteria we need are already on the plate. I'm going to now turn the plate about 90 degrees, and I'm going to slightly overlap the previous streak with this sterile loop. So what I've just accomplished is to take some of the bacteria from the first streak and I spread them out over the surface in the second streak, which was 90 degrees to the first streak. I'm sterilizing my loop again, not getting any more bacteria. And what I should have done the previous time, I'll do right now. Make sure your loop's cool. So touch the agar somewhere on the surface where there's no bacteria, turning it 90 degrees again and doing the same thing. I'm just overlapping a little bit with the previous streak and diluting the bacteria from streak number 2 even further in streak number 3. I'm going to sterilize the loop. Again, it's a little hot. I'm going to turn it 90 degrees again, and I'm going to cool the loop in the middle on the agar where there's no bacteria to make sure it's cool enough. And then one last overlap with streak number 3 to make a 4th streak. And you'll notice that time I overlapped a little bit with streak number 3, and then I did a sort of zigzag or fishtail pattern right across the middle of the plate where there were no bacteria to begin with. And so, again, the whole point is to create a dilute enough smear of bacteria that you'll get isolated colonies. And just to make sure this point is clear, let me demonstrate on the board briefly. So if this is your agar plate, what I did was I did my first streak of bacteria like this. And if this is my loop, I go flame it. And then make sure it's not too hot. I'll touch it in here somewhere. Then I'll overlap a little bit with the first streak like this. So this is going to spread out the bacteria a little more. Flame my loop. Do the same thing here. Flame my loop. Again, making sure it's cool in here. And then one final overlap and then drag it across the middle. And that should give a nice dilution of the bacteria. So in this area, you should get some isolated colonies. And that's the whole point of a quadrant streak.